In this video we will discuss about uh, in detail about the mechanism of this AOH1996, how it will cause DNA damage, whether it is through this TRC, that is transcription replication conflict, or through other ways. And in the previous studies of the same researchers or the same group has shown that this AOH will affect TRC in the pancreatic cancer cells. So in this video they will discuss the results, where they will go one step further to find out the mechanism that this AOH will cause the DNA damage. So first what it did is they used the PANC1 cell lines and checked the TRC in the control case without any AOH1996 treatment, and they are able to see that around 50% of the populations shows TRC. 50% of the population shows TRC. So how they detect this TRC? They check that this PLA which shows signals of less than 40 nanomolar. So they are checking the difference between RNA polymerase II and pCNA, that is proliferating cell nuclear antigens. So when they are close to each other, then there is a more possible probability of this transcription replication conflict. So they have find out the difference between, they have considered the difference between RNA polymerase II and this proliferating cell nuclear antigens. When it is less than 40 nanomolar, then there is a more chance of this TRC. So in the normal case, without any AOH1996 treatment, they see around 50% of the populations has TRC in the PANC1 cell lines. But when they use this AOH and with dose dependent increase in the AOH, they see a dose dependent increase in the TRC populations, indicating that this AOH has a role towards upregulating or increasing this TRC conflicts in the PANC1 cells. And same things they have validated using the flow cytometer, where we can see that when it's unstained, its signal is on the negative side, but when it is stained without an AOH, we can see around 50% populations are 50% of the populations are show TRC, and when we treat it with AOH, the signal shifts to the right hand side, it is high TRC conditions, and these things are quantified on the right hand side of the panel B, where we can see that between the control, that is DMSO, and AOH1996, the increase is statistically significant. And they also did their immunohistoning. Apart from the flow cytometry, they also checked using the immunostaining, where we can see that the difference where we can see that the mean PLA signals PLA is kind of this ligation assay, some kind of ligation assays, and here we can see that with dose dependent increase in the AOH, there is an increase in the PLA signals. And this is the immunostaining images, shows the increase in the TRC PLA within the cells. So within the nucleus, we can see that within the nucleus, the signals for this TRC PLA is low when it is 0 macromolar, but it increased with 0.5 macromolar, and it further increased with 2 macromolar, indicating that there is an increase in the TRC with increase in the AOH concentrations. Now they check the DNA damage in those cells using gamma H2AXP, and in this case they also use the transcription inhibitors as a control. So when in the control case, when there is only DMSO, they see a minimum DNA damage, and when they use the transcription inhibitors, the DNA damage is slightly increased. But when they only use the AOH, the DNA damage increased to a great extent, around 80%. But when they use AOH plus transcription inhibitors, the DNA damage goes down to the normal level where only DARB used, and this could be because of when they use the transcription inhibitors, the transcription of the DNA is inhibited, and this will prevent this transcription replication conflict conditions. And they also use one of the commercial drugs that is available for the treatment of the cancer, that is gemcitabine. And gemcitabine also increase the DNA damage, but when they use gemcitabine with transcription inhibitor, that is DRB, they do not see a decrease in the DNA damage, indicating that the gemcitabine does not cause DNA damage using TRC, but the AOH cause DNA damage using TRC. And these things they have did it also. The results on the right hand side of the panel D also shows the immunohistochemic images of the conditions that they did it in the, they checked it in the quantification of which they showed it in the left hand side of the panel D, where in the case of control they do not see too many TRC signals, but when they use AOH1996 they see an increase in the TRC signals. And in the case of this inhibitor, they do not see any TRC signals, and similarly with this AOH 1996 and these transcription inhibitors, they do not see any signal, or minimum, they see minimum signals for these transcription inhibitors. But when they use gemcitabine, they see more signals for the DNA damage, and when they use the gemcitabine with DRB, they still see signals for the DNA damage. So here they do not. So I want to correct myself that here the signals of the pink is not transcription replication conflict, but it is representing DNA damage, which is done by the staining of this gamma H2AXP, which is done by the dye, that is gamma H2AXP staining, which binds to the, which shows the DNA damage. So whenever there is DNA damage, this dye will undergo phosphorylation and gives signals. In the panel E, what they did is they check the AOH using the Western blot, and in this case they use two kind of AOH, that is they don't check AOH, they check the RNAP2, and which in the previous literature shows that with AOH treatment this RNAP2 degraded. So they use two different kind of RNAP. First one is the active, and second one is this RNAP2 inactive. And what they are able to find out is that when they use AOH, they are able to see a decrease in the active RNAP O, which is the active RNAP in the AOH conditions. But when they use this inhibitor, that is RNAP inhibitor, that is MG132, they do not see a degradation of the RNAP2. And similarly for the positive control, they use the UV, where when they use the UV, when they use only the UV, they can see degradations in the RNAP, active RNAP. And when they use the UV with MG132, they do not see degradation in the active RNAP because it is inhibited. And for the negative control, for the positive control, they use this inactive RNAP2, which does not get affected by any conditions. So they did studies in the PANC1 WC and MIA PACA WC. In both the cases, we can see that the RNAP get degraded with the treatment with the AOH, but does not get degraded when the RNAP is inactive or inhibited by either by these inhibitors, the MG132. Thereafter, they check whether this AOH treatment has any effect on the overall RNA translations or not. For that, they check this RNA fluorescence intensity at different conditions. When they use the DMSO, they see a high intensity, but when they use this DRB, that is transcription inhibitors, they see a decrease in the RNA fluorescence intensity as expected. And with AOH, as it increases this transcription replication conflict, we also expect to see a decrease in the transcription or decrease in the RNA fluorescence intensity. And for the gemcitabine, we see a slight decrease, but that decrease is statistically significant. And panel G represents the immunofluorescence images of the results that are quantified in the panel F, where we can see that in the case of DMSO, we see a high signals for this RNA fluorescence, but when they use the AOH1996, we see a decrease in the signals for the EU dye, and similarly for the DRB we see a decrease in the signals for the EU dye. But for gemcitabine we see a slight decrease in the intensity of the EU, but that decrease is statistically significant. So this all in together comes to a conclusion or summary that AOH1996 affects TRC, that is transcription replication conflicts, or in more precisely it upregulates or it increases this transcription replication conflicts in the pancreatic adenocarcinoma cells, and that is shown here by PANC1 cell lines and the this MIA PACA2 cell lines. So I hope you understood and somewhat convinced that AOH1996 affects transcription replication conflicts in a positive manner in the pancreatic adenocarcinoma. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.